Good afternoon, everyone. I'm Disanayaka, and I'll be presenting my research on first report of Curvularia chiangmaiensis on Rajarata in Sri Lanka. Here's the content of my presentation. Curvularia is a species-rich genus within the family Pleosporaceae. Members of this genus are mainly characterized by having curved conidia, which is resulted from asymmetrically swollen intermediate cells. And there are some species with straight conidia as well. According to the databases and previous publications, only 26 species of Curvularia have been reported so far from Sri Lanka. So the members of the genus are associated with various hosts, and also they can have various lifestyles as well. They can live as saprophytes, endophytes, and pathogens. Pathogenic species of Curvularia can cause various types of plant diseases, such as seedling blight, leaf blight, black kernels, green molds, and leaf spots as well. Most of the Curvularia species are known from poaceous hosts. When it comes to family Poaceae, they include many important, commercially important cereal crops such as barley, oat, millet, and rice as well. As we all know, rice is consumed as the staple food in Sri Lanka. When it comes to rice or the genus Oryza, this genus Oryza includes around 24 species across the world. But from that, only 2 species are cultivated. Those are Oryza glaberrima and Oryza sativa. Oryza sativa, or the Asian rice, is the one that we consume. Other 22 species are wild Oryza species. From that these wild Oryza species are considered as the ancestors of the cultivated rice. So these wild Oryza, from the 22 species of wild Oryza, only 5 species can be found in Sri Lanka. Those are Oryza nivara, Oryza ekingili, Oryza granulata, Oryza rhizomatis, and Oryza rufipogon. This Oryza rufipogon is considered as the wild progenitor of the cultivated rice, or the Oryza sativa. According to the references, these wild Oryza species are genetically stronger than the cultivated rice varieties. So the pathogenic fungi associated with wild Oryza species can become potential pathogens to cultivated rice varieties as well. But the fungi associated with wild Oryza species have been largely overlooked. To address this gap, my research study's main objective was to collect Curvularia species associated with Oryza rufipogon and accurately identify them using morphological and molecular data. Here's the overview of my methodology that I have followed. First, the symptomatic samples were collected from the locations listed in this previous publication. And the herbaria prepared for Oryza rufipogon were authenticated by National Herbarium Peradeniya. Fungal pure cultures were obtained using single spore isolation technique, and the pure cultures were maintained on potato rosetta, and stock cultures were maintained on corn vinegar. Morphological characterization was done based on the culture characteristics such as growth rate, color, texture, holly shape, and margin, and also micromorphological structures such as hyphae, conidiophores, and conidia. These are the major steps that I have followed in molecular characterization. DNA extraction was done using modified sodium tautosyl sulfate method. And the PCR amplification was done to both ITS and JPDH regions using these tailored primary sequences and the protocols. PCR products were visualized by agarose-gel electrophoresis using 2% agarose-gel, and the amplified products were purified and sequenced by Sanger DNA sequencing. These are the softwares that I have used in sequence alignment and phylogenetic analysis. Phylogenetic tree was constructed using newly generated ITS and JPD sequences together with selected X-type and reference sequences obtained from GenBank. Moving on to the research part, initial identification of the fungi to the generic level was done based on the microscopic characters. Accordingly, 6 isolates were identified in the genus Curvularia. After molecular characterization, one isolate 8017, was identified as Curvularia chiangmaiensis. Here you can see the details of the fungal isolate that I have collected, and this table shows the GenBank accession numbers of the newly obtained sequences. Here you can see the resulted maximum parsimony phylogram for the Curvularia species. As you can see here, isolate AD017 is clustered with the X-type sequence of the Curvularia chiangmaiensis. Here you can see the culture characteristics, some of the culture characteristics and microscopic images of the isolate that I have obtained. And this Curvularia chiangmaiensis has been recorded from various hosts, such as Saccharomyces officinarum, Siamese, Oryza sativa, Panicum virgatum, and Penicitum pediculatum. In conclusion, I can say that Curvularia chiangmaiensis, isolated from Oryza rufipogon, updates a novel host fungal association from Sri Lanka. This fungi have the potential to switch their life modes from saprobic lifestyle to mild pathogens, and in some cases to severe pathogens as well. As I said earlier, this Oryza rufipogon is considered as the wild progenitor of the cultivated rice, or the Oryza sativa. So even though the pathogenicity of the species collected from Oryza rufipogon described here are not confirmed yet, they may have the potential to cause severe diseases in Oryza sativa by switching their life modes in the future. As future works suggest that it is important to test whether the identified fungal isolates can infect cultivated or other wild grasses to understand their potential spread. And also, since wild Oryza species may host pathogens that could infect cultivated rice, their management and monitoring are important in disease prevention. And moreover, it is important to establish surveillance programs to monitor changes in fungal populations and emerging pathogens in wild Oryza species. Here's the acknowledgement, and these are the references that I have used. Thank you.